Welcome, everyone. A very warm welcome to this first alumni day at the University of Southampton, it's first in my experience, and particularly to this set of small talks, lectures we're offering on the future of medicine, smart partnerships. I hope you'll find it really interesting. Certainly we've got a great group of speakers to talk to you today, and hopefully plenty of time for you to ask questions and hear some answers in due course. I'm Mark Spearing. I'm the Vice President for Research and Enterprise at the University of Southampton, and it's my pleasure to be your host for this event this morning. This is a fantastic day. I hope it will become a very regular event. It's about sharing memories. It's about celebrating the 70 years that we've been a university with a charter, the 160 years we've been an institute of higher education here in Southampton. It's about meeting old friends, meeting some new friends. It's about looking back. But what we're celebrating here in what we're offering in this particular session is to give you an update on what we're doing currently in a really important area around health and looking a little bit into the future about what we might be doing over the next decade or so. And it's the future of health, future of medicine rather, but smart partnerships, and offering to you that the way we need to do this is by working in partnership, working in an interdisciplinary way, bringing people and ideas together that haven't been brought together before to develop the synergies between them and see what emerges in terms of what might be possible in the future. I'm the Vice President Research and Enterprise, and it is the best job in the university. Of this I have absolutely no doubt. I'm responsible formally for ensuring the university has a strategy, a framework within which people can do amazing things. To think creatively, to work together in unexpected ways, to produce brilliant ideas, sometimes that are ideas just for their own sake, that are thinking in ways that people haven't thought of before. Things emerge that are just fascinating, fantastic. But sometimes it's about bringing the right teams together to tackle difficult societal challenges, global challenges even, to make a difference. And both are equally valuable and everything in between. It matters that we as a major research university do that. This has been a good week for the university and people in my job, or my job in particular, which is we received our Research Excellence Framework outcome, and it was very positive. We've gone up in that ranking. We're now the 14th ranked university for quality in the UK. Particularly pleasing was that we're now, have gone up really significantly, we're now the 7th ranked university for the impact of our research in the UK. And that's very relevant to what I was just saying about addressing societal challenges, whether that's global health, global hunger, the nexus of food, energy, security with water security, aging populations, urbanization. These are things that are big challenges for us as a global community. And the only way one can address those is by working in an interdisciplinary way. I'm an engineer by background. There's a conceit that bring great technology and it will provide the answers. But my lifetime of experience says that you can have the wackiest, whizziest technology that provides medical diagnosis or makes your car run more efficiently. But if people don't use it, if it doesn't work within culture, within a regulatory policy framework, it's worth nothing. It's not going to happen. And if it doesn't work economically, similarly. So it's really, really important that as an individual university, as a research sector, that we work in an interdisciplinary way, particularly when we're looking at addressing challenges. And quite frankly, there are no bigger challenges that face society than around the challenges that medicine, healthcare, public health, long-term global disease, and if you're in any doubt about that, just think back over the last couple of years and what COVID has done to us as a society, and the relative neglect of vaccines and infectious disease, how we've had to rally together to address that. And I'm very proud of the contributions that this university has made to the vaccines program, to the saliva testing program, the PROSECO program that was very interdisciplinary in bringing our engineers and medics together to, in 5 days, to come up with a workable, manufacturable design for a forced air respirator using commercial off-the-shelf parts, and an interaction between a professor of respiratory medicine, Paul Elkington, Hal Morgan, an engineer who works in the biotechnology area in particular, allowed that to happen. So we have a really good track record in this space. And what we're going to hear about today is a couple of areas that I think are of particular interest to many in the room: cancer and dementia, neurodisease, and giving you a sense of how we're going about addressing them now and what the future might look like by partnerships between medics, clinical medics, and engineers, biologists, imaging specialists. And I look forward to hearing what they have to say. To chair the first session, which is on cancer immunology, I'm going to hand over to my esteemed colleague, Diana Eccles, who is the Dean of our Faculty of Medicine, is a practicing clinical academic herself, and a professor of cancer genomics. Diana, over to you. Thank you.
Welcome everybody. So I'm Diana Eccles, I'm Dean of the Faculty of Medicine. I'm a Professor of Cancer Genetics, and previously, before 2018 when I took over this role, I was actually Head of School for Cancer Sciences. So at the start of my time as Dean of the Faculty of Medicine, it was exactly at that point in time that we opened the Centre for Cancer Immunology, which many of you may well have been involved in, and where you will probably all be aware that it was entirely philanthropically funded. So we're really, really proud of a very beautiful building, but even more proud of the staff who have populated and used that building to further our fantastic cancer immunology research. And we're very lucky to have one of those individuals today talking to us. We were able to recruit in, with this wonderful building and the great facilities we had, two very talented researchers, very definitely interdisciplinary, to work in that building and grow a group. And Sally Ward, who's one of those individuals, was just this week awarded the Fellow of the Royal Society, which is one of the highest accolade in science in this country. So we're really, really proud of Sally and really delighted we've managed to get her to come to Southampton. We've got big plans for the future for the Faculty of Medicine, and plans to really improve yet again another step change in the facilities that we offer our brilliant researchers to work in. And neurosciences is one of the other areas that we're really wanting to push more and more to the fore in the future. And it already, we have some great excellence in that area, and you'll hear from one of our staff and her teammate. Now I say teammate because both our speakers today from the Faculty of Medicine are speaking to you with colleagues in another discipline with whom they are working to really push the frontiers of their science. So I'm going to hand over to the real entertainment, and our first speaker is Dr. Sian Lim, who's from the Centre for Cancer Immunology and is a clinician and clinical scientist, and then Owen Rackham is a computational biologist who will follow Sian. So thank you.
Good morning, everyone. It's a real pleasure to be able to speak to you today. And as Diana's just introduced, I'm Sian Lim, and my job title is a mouthful, but what it means is that I am a consultant hematologist working in the NHS. I specialise in blood cancers, especially lymphoma, and I'm also a research group leader based at the Centre for Cancer Immunology, where I'm interested in finding new cancer treatments. And today I'm going to talk to you about our research, where Southampton led, in COVID-19 vaccine research in patients with blood cancers, particularly lymphoma, and how might this information help us design new cancer treatments. So here's an introduction to blood cancers. Blood cancers happen when the stem cell, a baby blood cell, the developmental pathway goes wrong. It's interrupted, and this leads to accumulation of a particular type of cell. In this case, if it's a B cell, a type of immune cell that has gone wrong, then this leads to cancer. And this might manifest in patients in the form of a lymph node swelling, as seen here in this patient, where on this scan, and unfortunately in this case, this lymph node swelling is compressing the patient's windpipe. So early in the few months into the start of the pandemic, a research group studied the health records of 17 million patients across 17 million individuals across the UK. And what they found was that patients diagnosed with blood cancer are 3 times more likely to die from COVID-19. And this was pre-vaccination. And it doesn't come as a surprise because blood cancers are essentially a cancer of the immune system, and we need the immune system to protect us against COVID-19. So how does your immune system protect you against COVID-19? Well, this is a cartoon of the SARS-CoV-2 virus that causes COVID-19. And the virus needs to enter our cells to cause COVID-19. The virus, and it does so by a lock and key system. So on the virus are these structures called spike proteins, and that's the key. It needs to bind to the lock, which is the host ACE2 receptor on our cells. And when it does so, it is able to open the door into our cells. Now, the immune system can counter this, can protect us by producing antibodies, and the antibodies bind to these spike proteins so that it can't bind to the lock. And in doing so, it means that the higher the antibodies, the better. And we believe if we have high levels of antibodies, then it protects us from getting infected in the first place. There's another way the immune system can protect us, and this is through T cells, which is another type of white cell. And what the T cell does, if it's able to it is able to recognize a virally infected cell, then it can directly kill and destroy the cell. And these T cells, they're really important for reducing the risk of hospitalization, for reducing the severity of COVID-19. And in December 2020, the government introduced the national vaccination program. And across the country, as blood cancer doctor and a researcher, we were all immediately worried if the vaccine would induce a sufficient enough immune response to protect our patients. To answer this, we designed the PROSECO study over a glass of, yes. Now, PROSECO recruited many different types of lymphoma patients. I'm going to focus on one group, which is patients with indolent B-cell non-Hodgkin's lymphoma. In a 5-year period, about 65,000 people in the UK have this condition, and it is a slow-growing sort of lymphoma, may not need treatment, and we have lots of treatment options, but it is never curable. And so what we did was we recruited 500 patients to our study. We grouped them into patients, the patients depending on whether they had cancer treatment or not. And then we took blood from them before their first dose of vaccine and after each dose of vaccines up to 3 doses at the time being. And the aim really was to evaluate how good the immune response to vaccination was, how long this immune response lasted, and really, could we predict who is going to be a good vaccine responder? And at the same time, we had a group of healthy volunteers that we used for a comparator group. So how do vaccines protect? Well, both the AstraZeneca and the Pfizer vaccine, and lots of other vaccines for that matter, they contain instructions for the cells to make the spike protein, so just the spike protein. And when injected, another type of immune cell called dendritic cells take up the vaccine and they make the spike protein and express it on the surface of the cell. Now, this means that when B cells, if the circulating B cells in our body are able to recognize the spike protein, then it changes into an antibody-producing cell and produces lots of antibodies against the spike protein. But also more importantly, this cell then becomes a memory B cell. And as the name suggests, it's really important so that in the future, when we encounter the virus, it can respond quickly to produce more antibodies. And so we can measure the immune response to the vaccination by measuring these anti-S antibodies, and the higher the value, the better. So here are two graphs of results from the study. And just to orientate you, the y-axis shows the antibody level. And here we have patients who we've checked the antibody levels before their first vaccine dose and then after 2 vaccine doses. And these are all the lymphoma patients who were vaccinated whilst they were receiving cancer treatment. These are lymphoma patients who received the vaccine when they were not on lymphoma treatment, and these are healthy donors after 2 doses. Now, and the grey area means no detectable antibodies. Now, immediately you can see the healthy donors have a nice, clean response. It's really variable between the lymphoma patients and, most significantly, the patients that were vaccinated whilst they were receiving cancer treatment had a far lower response than the ones who were not. And this response was, this pattern was the same regardless whether for the AstraZeneca vaccine or the Pfizer vaccine. So some 50% of patients who were vaccinated had detectable antibodies compared to close on 100% that didn't. Now, but this variation was interesting. And so we asked what was making the difference in patients who were not on cancer treatment. Why were some responding and why some others? We asked first, is it a difference according to the disease? And when we looked at the responses based on their lymphoma type, you can see those patients with a slow-growing lymphoma, many of them, in fact, have rather low antibody levels compared to other groups. And this was after 2 vaccine doses. Now, after the 3rd vaccine dose, we saw an improvement in response, but still, many of them still have relatively low antibody levels. Now, it's complicating, I apologize, but what I want to say here is about the T-cell response. The vaccines also induce a T-cell response. Now, you remember the dendritic cell, it's got the spike protein on the surface. Now, it can also be recognized by T-cells, but for it to do that, the spike protein needs to be chopped up into little fragments by the dendritic cell, and these little fragments can be recognized by some T-cells. And if the T-cell recognizes it, then it divides to become an effector cell, and this effector cell is a killer cell that can kill the virally infected cell. But like antibodies, like the B cells, it can also become a memory T-cell, also very important to protect us in the future when we encounter different variants of the virus, for instance. And so, for the study to measure the T-cell response, what we do is take the blood, take out the T-cells, we add fragments of the spike protein, and we measure the ability of the T-cells to produce a chemical called interferon gamma. And so we measure the level of interferon gamma. Another graph, a y-axis, again, same principle. This is the interferon gamma level, and so the higher the response, the better. Each circle is a different patient. The shaded area is no response, and anything above the gray area is good. You're looking at responses after 2 doses in this graph, and this is the responses before vaccination in healthy donors and in patients lymphoma who were vaccinated whilst they were receiving cancer treatment and if they weren't receiving cancer treatment. Unlike the antibody responses, you can immediately see there is no effect on cancer treatment, meaning patients are producing an antibody response, a T-cell response, regardless of cancer treatment, but there are a good number that have no T-cell responses. When we looked at the response after the 3rd vaccine dose, and here we looked at people who did not respond after 2 doses, we can see reassuringly the T-cell response improves. And in those who had responded after 2 doses, they held or boosted their T-cell response. So that 3rd dose is really important. So here are the key findings of PROSECO. We found slightly under half of patients with lymphoma who had an antibody and a T-cell response, which is fantastic. Unfortunately, the blue section tells you that some 10% of patients with lymphoma do not have an antibody or a T-cell response, and this is a fraction of people that we are worried about. And then a third only have an antibody response, and about 20% have a T-cell response only. Now, what this study did was identify that some patients with lymphoma are not protected by, or we fear are not adequately protected by vaccination, and they are people who are undergoing cancer treatment, and they are people who have an indolent lymphoma at all stages. So whether they are on treatment or not, we think they might be at risk. And this data was used to guide the third dose, the decision for the third dose offered by the government, and also used to guide the vaccine task force as well as the JCVI to decide who should be eligible for COVID-19 medicines in immune-suppressed individuals. It is also one of the largest studies. It is also one of the largest studies internationally for lymphoma patients examining COVID-19 responses. Now, so this is all great, but how does vaccine respond? How is it relevant to cancer treatment? Well, the immune cells that fight infection, that fight virus, are the same immune cells that fight cancer. We're all developing cancer. Our cells are constantly making mistakes. The statistics is 1 trillion mutations a day. That's the number of mistakes our cells are making. And it's trying to form cancer all the time, very, very disconcertingly. But the fact is, it doesn't, because our immune system is really good at killing rogue cells. Until, of course, it doesn't. And the cancer cell manages the means of suppressing the immune system and multiplying. So the question is, how do we reawaken and re-engage the immune system to control the situation and to kill the cancer cell? Now, in PROSECO, we have collected blood samples from 500 patients, multiple time points. This allows us to track the cancer cells, how it goes from A to B by measuring the genetic information. And this is where the next speaker, Dr. Owen Rackham, will come in with his computer science knowledge. Now, by using the comparison between someone who is responded to the vaccine versus someone who hasn't, we believe that we can use this information really to unlock the clues to re-engage the immune system to fight both infection as well as cancer. This brings me to my last slide, and like I said, this is one of the largest studies internationally for lymphoma, one of the most detailed in terms of assessing immune responses. And the study was done in a time when hospitals were in lockdown, research teams were shifted to support the clinical service, and patients were asked to shield. So the fact that I can stand here today and present you this research, it's really because of the goodwill and hard work of these individuals. And we're really fortunate in Southampton to be supported by an experimental cancer medicine center that can produce this really high quality, tightly controlled data, a clinical informatics research unit that built us a database when I realized the Excel sheet was really not good enough, a clinical research facility, a trials unit with a fabulous statistician, the Center for Cancer Immunology, where there is a lot of additional expertise. And clearly such a trial is very much a national effort that involves multiple partners from across the country. And finally, obviously, a big thank you to the participants and the funders. So thank you for your attention. I will finish here, but I will now like to introduce the next speaker, Dr. Owen Rackham, to explain how we get from A to B. Thank you.
Thanks, Jan, and it's a great pleasure to get the opportunity to talk to you all this morning. Hopefully we can go to the next slide. So I want to tell you a little bit about how we're going to move on from this result that Jan has just described to you. And I want to tell you a little bit about how taking an interdisciplinary approach to these problems that emerge can help us really sort of push forward the boundary in terms of how we can make discoveries in medicine. So I wanted to start by a little bit saying something about my view on interdisciplinarity and how it was that Sian and myself came to work together. So I am first and foremost a computer scientist, actually. I trained in artificial intelligence. I cut my teeth actually working out how best to send trucks to put salt on the road when it snows. So you can see I've moved slightly on from then. And in a journey that I can't tell you in 10 minutes, I now am also considering myself a developmental biologist. I'm interested in stem cell biology and how organisms develop. And I hope you can see that this puts me slightly in a different place to Sian, but I'm going to show you some technology that my group's worked on, and hopefully you'll be able to see why working together lets us tackle problems in a slightly different way. So this is my view of the world. Often we collect data, it makes us realize that there are some problems, and then we're going to use technology to try and find a solution to those problems. So actually, I'm really focused in on that section. What are those technologies? And the nice thing about it is if you're working in an interdisciplinary way, you don't actually always need to come up with new technologies. Actually, you just need to realize that something that you've made in one area, you can use it to make progress somewhere else. And hopefully, what I'm going to show you in the next few minutes, is an example of how Sian and I are trying to do that. This arrow is very important because the great news for us is that often when we come to a solution, that actually just puts us back at a new problem and we end in this cycle, which means we always have something new to work on. Okay, so in my computer science view of what Sian just told you, this is kind of the situation that we're in. We have non-responders and we want to make them responders. Okay, so I work in flowcharts, so that's my understanding of this problem. Okay, and I'm going to talk to you about a technology, not for this, but for a very similar problem, and then hopefully you'll be able to see why it is that Sian and I are so excited to be able to work together. Okay, so human biology is amazing. We have a whole host of, oops, that's not the pointer, this is the pointer. We have a whole host of different cell types that make up our bodies, and they all arise from initially this single cell that divides, and as it divides, it becomes, it specializes into all of these cells. And this is truly a remarkable process, and I talk to you as a father of a 5-week-old daughter, so I really know the sleep-depriving beauty of this process. But the interesting thing from my point of view is, how is this possible? How is it that one cell type, this stem cell, can give rise to all of these other cells? And how can we use that to help us innovate in terms of how we can use that process to do new things? Okay, so the sort of potential of this changed a lot, and I'm going to show you one experiment that really demonstrates how our view of biology can change over time. So we often use this analogy that a stem cell is somehow at the top of a mountain, and then during development, it's going to roll down this mountain, and as it goes lower and lower, it becomes more and more specialized, and it ends up in all of these different cell types that we use in our body. And for a long time, we thought that that process was a one-way process, that those cells at the beginning of our life, those stem cells, they are just going through an ongoing process of specialization, and once they've reached their final destination, that's where they'll stay. But I'm just going to show you one experiment. There are several, but I think this one is a very clear example of how we learned that wasn't the case. So this is Harold Weintraub. He did this very clever experiment. So if you take a fibroblast, which is actually just a cell from your skin, and you put it under the microscope, it looks like this. What that cell type does is it does a lot of things like creating scars. It provides a lot of the structure for your skin to grow on. But the amazing thing, in my opinion, and I hope you agree, is that if you take that fibroblast, that skin cell, and I'm going to put into it 4 things. They're called transcription factors. You can think of them as the coordinators of your cell. These are the genes, the proteins, that coordinate which things are being used in which different cell types. So I'm going to take 4 transcription factors, the sort of politicians of the cells, if you like, and I'm going to put 4 of those into a fibroblast that wouldn't normally be there, okay? And then I'm going to wait for a while, and I'll put those cells back under my microscope. And this is what happens. So these are the same cells. I'll go to the next slide so you can see a bit more clearly. These are the same cells after about 4 weeks, and hopefully you can see that they're beating, okay? And that's because just by adding those 4 transcription factors, those coordinators of behavior, I've been able to take those fibroblasts and turn them into cardiomyocytes. So these are cells from your heart, okay? So this and other experiments really demonstrate that actually this mountain idea is not the case. We can probably move freely around this landscape. The difficult part is knowing how to move around that landscape. What I didn't tell you is what Harold did was actually, he came to that discovery by accident. And then a lot of other experiments, they've taken an awful lot of time to try and find the different sets of transcription factors to move. So we know we have this landscape. Humans have about 400 different cell types, and we want to be able to navigate it. But every different conversion, so that would be from 400 to 400, which is a very big number, requires a different set of regulators. So you can see that if we want to navigate this space, we can't find the right transcription factors, we can't find the right regulators, either by relying on luck or by relying on trial and error. So that's where computer science can come in. That's where machine learning can come in. That's what I just said. And so my group, and I'm afraid in 10 minutes I can't tell you too much about how it works, we developed this technology called Mogrify. And basically, Mogrify is a technology that helps us answer that question using computer science. Okay? So we can take any cell type and predict what regulators you have to put into it so that it will become another cell type. Okay? And I shan't talk about the steps today because I'm a bit worried I'm going to run out of time. But you can think of this a little bit like we're trying to start a revolution, right? We have one cell type, our fibroblasts, let's say, and we want to start a revolution in that cell type so that it becomes a cardiomyocyte. So we need to pick the leaders who are the best connected and have the most influence so that I can convince, if you like, my fibroblasts to become cardiomyocytes. And it's that logic that this machine learning algorithm, Mogrify, is going to use. Okay, so you can think of it like this. We have any human cell type, we apply this algorithm, it's going to tell us what we need to do so that that cell type can become any other cell type. So I'm sorry, I live in the world of analogy. This is where we want to be. We want to be able to stand in this landscape and look at a map. Unfortunately, we live in England, and in the world of cell biology, this is also what it looks like. We're trying to navigate this landscape without being able to see anything. And we're trying to do this. We're trying to develop this GPS system for exploring space. So I'll just show you a little bit how this looks right at the front end. Basically, you can come, you can say, like in a travel website, if you like, you say where you want to start, you say where you want to end. That algorithm is then applied to a whole bunch of data, which I didn't have a chance to tell you about today, and it predicts these are the transcription factors that you need to do this here. My aim is not so good with the pointer. But hopefully what you can appreciate is that this is a huge problem that we need to solve. It's too difficult to solve just by taking a trial and error approach, but by using a computer science approach, we can try and solve all of those problems in one go. Okay, I'll just show you very quickly an example of this. So a keratinocyte, which is another cell type in our skin, we asked the algorithm, how do I convert that into a microvascular endothelial cell, which is the cell that make up the small vasculature in your body. The algorithm predicts how to do that. I overexpress those, and then these cells become microvascular endothelial cells. So that is a technology based in computer science that we're using in developmental biology. And then I'm fortunate enough to meet Sian, and hopefully you can start to see some similarities in the problem that Sian puts forward, which is how do I convert a non-responder to a responder, and how that mimics the same kind of questions that I'm asking here. How do I turn a fibroblast into a cardiomyocyte? So this is the movement across the landscape. So this is really a problem that Sian and I are working on right now. So we can take the technology, we just have to change it slightly, and I just want to try and give you a little bit of a flavor of how that works. So in my world, I'm trying to go from a fibroblast to a cardiomyocyte. And these are two very, very different cell types. And actually, in some senses, that makes the problem easier, because spotting the differences is easier when you have two very different things. In the problem that Sian puts forward, we're actually more here. So we now have to try and work out how to modify this technology to spot differences between things that are very, very similar. And then we hope that we can use that to be able to convert the non-responders into responders. And that's the end. You'll have to come to the next alumni day to see how well we managed to apply that. And thank you for your time. And both of us will be happy to answer some questions.
So with that, we're going to move from cancer immunology, to dementia. And it is a great pleasure to welcome Roxana Carreri and Sumit Mahajan, who are going to talk. Roxana is going to talk first. Roxana is one of our leading biomedical scientists in dementia studies. Roxana is an outstanding individual as well. She was the honorary consul for Romania in Southampton, and she introduced me to Dame Esther Ranson, which was a really interesting experience. Roxana, dementia.
Thank you very much. It's a great honor and pleasure to be here, and the reason that I met Dame Esther Ranson is the same reason that I'm enthusiastic about this topic. We are all aging, and quite frankly, as an anatomist, the alternative of not aging is not very nice. So what happens with aging is that the age-related diseases, their risk increases. And the number one risk factor for dementia is aging. There are other risk factors that are usually associated with cardiovascular health as well. So dyslipidemia, high cholesterol, there is a certain genotype that predisposes to dementia as well. The question is, and the reason that there is no efficient treatment for the 850,000 sufferers of dementia in the UK, is because the causes aren't exactly clear. We know the brain is enclosed in the skull, so it's inaccessible. We need good imaging, and you're going to hear more about imaging techniques from my colleague Sumit Mahajan. What you see in a demented brain is, first of all, there is loss of brain substance. So the brain becomes atrophic. It's smaller. The cavities that contain cerebrospinal fluid that we need in order to bathe the brain, to feed the brain, those cavities enlarge. And that's usually a bad sign because it's a sign of the fact that you're losing brain substance. The question is, why? Why does this happen? Obviously, we need to do something about it because it's a huge societal and economic burden. There are two major things that happen in the demented brain. One is accumulation of proteins inside the cells, and the other one is accumulation of proteins outside the cells. The problem is that we all produce these proteins. These are not mutated proteins. We produce them. So the question is, why is it that a young brain can cope with clearing, with eliminating these proteins, and an older brain, some older brains, cannot get rid of these proteins? And this happens. They build up in the brain as amyloid beta sticky plaques, you may have heard of them, or tangles within neurons. Now, there is one major anatomical fact that is peculiar about the brain. It does not have lymphatic vessels. So everywhere else in the body, there are vessels through which fluid, cells, immune cells, proteins can drain away from the tissue, and they can be cleared from the tissue. The brain and the eye are the only two organs that do not have these lymphatic vessels. So the major question that I asked myself about 15 years ago is, could it be that Alzheimer's disease is a consequence of not having efficient lymphatic drainage? And there must be lymphatic drainage in a younger brain, so what are the lymphatic drainage pathways of the young brain? Because if we can identify them, if we know the drivers of efficient lymphatic drainage in the young brain, we can do something about the older brains. So, can we have the lights down, please? Because this is, I think, the key to what I'm about to discuss. Technology is a wonderful thing. And when we took the brains from people with different stages of dementia and analyzed them carefully, we noticed that there is the degree of dementia associates very closely to how much amyloid builds up in the walls of the blood vessels. So what you can see here is a vessel from a demented brain that is in longitudinal section, and this is a cross-section of an artery at the surface of a demented brain. Now, an artery has an inner lining, it has a middle lining, and in this middle lining sheet, there are smooth muscle cells. Their role is to pump the wall of the artery. And it is around these smooth muscle cells that amyloid builds up, always. Now, we're very fortunate in Southampton because we have a lab where we've got anatomical specimens, we have a brain bank, so we can do these studies. And across a number of experimental studies and hundreds of brains that were examined, we managed to define and characterize that the lymphatic drainage pathways of the brain are along the linings of the blood vessels, and most importantly, these linings that act as cement between the smooth muscle cells. As we age, the structure of these linings changes as well, and therefore you're more prone to accumulating amyloid beta. But here's the problem. We're all aging, we all change the structure of our extracellular matrix and these linings. Why is it, we can put the light on now again, thank you. Why is it that this process that we called IPAD, by the way, it's intramural periarterial drainage. It happens only along the walls of arteries. Why is it that this IPAD process fails in the aging brain? Well, one major question that we had, where we needed everybody of every specialty in the university, and I think I'm hoping that I'm going to convince you with the next slides, is what is the driving force? What is the motive force for this lymphatic drainage along these pathways in the brain? And for this, we had to team up with physicists, a vascular surgeon, a cardiovascular surgeon, a medical physicist, and a mathematician, and there were computer scientists there as well with us. And the reason is these pathways are very inaccessible. You can't see them, you can't reach them. So what we did was we formulated a hypothesis, and our hypothesis was that it's the pulsations from the heart that drive IPAD. And of course, the mathematician said, yeah, but the heart's down here, your brain's up here, they're in the wrong direction, it can't work. Within a year, we managed to actually find the answer to our question, and that is this strength and the amplitude of the contractions of the smooth muscle cells themselves that drive this lymphatic drainage. And this, of course, for us is wonderful news, because the smooth muscle cells have sensors on them, these sensors are innervated and we can act therapeutically. So we now have 2 major programs of work towards treatment of Alzheimer's disease and facilitating this process, IPAD. One program of work is to use chaperone molecules, such as this one's called apolipoprotein J, so it helps with the transport of cholesterol. It also helps with the transport of amyloid beta along these pathways. And our collaborative work with the Mayo Clinic in the United States has shown just that in an experimental model. So we're working on that side as one direction. The other direction, which is very exciting, but it's a little bit more complicated, is that these smooth muscle cells are innervated. And the innervation comes from 2 major centers in the brain. These 2 centers get tired with aging, so we need to fire them up again. And you can do that by delivering drugs that they would have delivered had they been younger and healthier. So with this, of course, there also comes, well, what about the smooth muscle cells themselves apart from the sensors? Maybe we can restart their machinery, their ability to contract, and for that, work on the mitochondria, which are the energy-producing parts of the cell. And there are drugs that are already approved that act on mitochondria in other cell types. So we're going to try them in the brain, and this is work that's, again, collaborative with the United States. So right now, as opposed to when you met Dame Esther Ranson, we actually have some targets for therapy. This is a very exciting time for us as an interdisciplinary team because we're no longer trying to find what goes wrong. We are working on therapeutic interventions. One of the major problems with Alzheimer's disease is that it's diagnosed late. What I showed you at the beginning in the brain appears 20 years before the symptoms start. So it's also key to try and find some very reliable biomarkers. So I mentioned the fluid in the cavities of the brain. Well, work that we are doing, again, at an interdisciplinary level in Southampton with ENT surgeons is showing us that that fluid drains in very small quantities towards the nose. So we might be able to detect the changes in that fluid that indicate Alzheimer's disease by observing the changes in the nasal mucosa. This is a massive international collaboration, and I'm very proud that me, as a scholar of Southampton, is actually leading an international effort that we call CLEAK. You see, I like my acronyms. This one's Clearance of Interstitial and Cerebrospinal Fluids, that is funded by the Alzheimer's Association in the US. Thank you very much.
Thank you, Rox, and it's a pleasure again to be here and talk about some of the work which we are doing in this space of dementia. So I'm going to actually follow on the act, which is a tough one to follow, but or to tell you about the work which we have been doing, stemming from going from very fundamental research to all the way to actually solving this problem of early detection of subtype in neurodegenerative diseases. So my name is Sumit Mahajan, and you'd be surprised, I am actually a chemist by training, but also trained then as a biomedical engineer. And what I currently develop are imaging solutions for biomedicine. It has quite a bit of a journey for me as well in terms of traversing different disciplines. But now I find it hugely exciting that actually I'm able to bring them all together and contribute to something which can actually have real world impact. So what I'm going to actually talk to you about are some novel technological solutions which have come out from our lab for personalized medicine and for improving patient outcomes in the space of neurodegenerative diseases. So, but first and foremost, I'd like to acknowledge the work, the collaborators with whom I've been working on it, and especially, and I could like to point out again the huge interdisciplinarity which you might see in the team, where we have tire biologists, we have people who work on photonics, which is my group largely, people in clinical neurology. So this is Professor Christoph Kipps at the University Hospital Southampton, and for nanocharacterization work, we have Dr. Iris Nandakumar. I mean, a key person I would point out here is George Devitt. He's now a postdoctoral research fellow who works across the two groups in tau biology and photonics, who has been hugely instrumental in bringing these two very different disciplines together to actually take things forward. And I'm obviously very, very thankful for the funders which have supported us, including the benefaction from Mr. John and Mrs. Elizabeth Bolden. What I'm going to actually show you now is some of the Rox mentioned that actually there are deposits which happen in the brain. So if you manage to take those deposits and put them on a surface and try to image them, this is how they look like. They look like these tangles. So Rox mentioned these intracellular proteins which accumulate intracellularly. So at the nanoscale, they appear like this. So although this is titled as Cyber Canyon because we submitted this to a micro imaging competition and got the first prize, but this is how they look. Yeah? And I'm going to tell you more about these in a bit. So the fundamental issue in all these neurodegenerative diseases, including Alzheimer's, is the fact that there is protein accumulation or aggregation, as I might refer to it. And that really starts here. You have the proper functioning monomer, the protein in its own. It undergoes a critical event called misfolding. It changes its structure somehow. And then it gets the propensity to actually start to accumulate and become assemble into oligomers, protofibrils, and fibrils. And it's the fibrils which actually deposit in the brain and as plaques or tangles. And that happens really as Rox mentioned, 20 years before this process actually starts. So what would be key to being able to early detect or detect things early would be able to detect this change and the triggers to this change. And that is precisely what we want to do. So these protein aggregation is called by structural changes, and if you could intervene in this stage, you could reverse it. Currently, there are no disease-modifying drugs, but because we can't catch things early. If we could do that, it would have to be at this stage when they still can move backwards and forwards. Here, it is permanent. It is the brain's way of sequestering things and kind of trying to say what it can. So before that, I'm going to give you a little bit of a primer that proteins have a hierarchical structure. They have a primary structure which consists of their amino acids stringed together as a thread. That is important. But a protein's most important thing for a protein for its function is a secondary structure, or how they arrange in space, whether as a helix or as a sheet or just floating around. And it's this secondary structure which then you can build upon together. There are superstructures on that which assemble together as tertiary or quaternary structures, but actually it's the secondary structure which is the most important thing. And if pathological change in this secondary structure is misfolding, and that's what we target in our detection technologies. So this leads to me defining the challenges which we have worked on and which our technology is able to address. And one of them is the fact that if we could detect these structural changes during accumulation, then we could actually develop structure-sensitive drugs. If you could work out what this pocket looks like, then we could exactly target it. And that could perhaps accelerate the search for disease-modifying drugs. The second aspect is that there are currently no technologies, no techniques, no methods which can do detection early, 20 years before. That's our target. Yeah? And if we had a method which could detect this protein structure at the molecular level as a disease marker in biofluids, then job done. That would be perfect. And that's exactly what we have been trying to do in our research. So we have developed a new laser-based technology, which comes from very fundamental research, because I mean, this technology, I'm a spectroscopist and an engineer by background, and we just were trying to play and make the technique better, more sensitive. To give you a very brief primer of the technology, it is laser-based. All our molecules are connected have atoms which are connected by chemical bonds. They're vibrating, and we detect that vibration by shining a laser light on it, and monitor what comes out. The scattering which the molecule does is what we detect. This is based on a technique called Raman spectroscopy, but you don't have to remember the name. But what we get is a chemical fingerprint. It's a signature of the material. We innovated on it by saying, okay, can we do it twice, thrice? And if we do that, we get better specificity. And we developed a workflow where we just collect biofluids from a patient, do a very simple sample preparation, no addition of reagents, just smear it on a slide, just put a drop on a slide, put it under the microscope, shine laser on it, and there you go. You get to identify whether it's a monomer at the early stage, or is a misfolded entity, or it is the fibril. You can do that already. And that differentiation, identification, classification allows diagnosis. That can mean that you can actually start taking, managing care, the treatment in good time. Yeah? So these are some results which show that actually your method works. So tau, the name of the protein which actually assembles intracellularly in Alzheimer's disease is called tau. And you can see by applying a method, when it goes from monomer, where actually there is this nanoscopic, and when it becomes oligomer to fibril, you can actually separate them out almost with 100% accuracy. We can also detect mutants. So if the tau protein structure is inherently defective, you can also detect it very well. So these are different isoforms of tau protein, and they separate out really well. We have developed then, based on the same strategy, we developed a stratification strategy for patients so that we can actually subtype disease. So when a patient shows up with some aspects of a neurodegenerative disorder or dementia, then the first thing that the clinician wants to know, is it really neurodegenerative disease, or is it really? And then within that, they want to know, well, how fast do I need to administer treatment? What can I do? And some of you might know that within Alzheimer's, there's a lot of heterogeneity. There is early onset disease, there's late onset disease, there's severity levels with different severity levels, and we want to subtype them so that we can actually differentiate them. We also checked our method against PSP, which is progressive supranuclear palsy, which actually is caused by the same protein. It has some overlapping symptoms, so it's very important to actually differentiate between AD and PSP. And our method was we tested our method on patient samples. These are CSF samples from patients, and each dot is a patient, and you can see all of them, we get very, very nice results. Almost, again, 100% identification. That we can say even though they might have overlapping symptoms, we can actually tell AD patients completely apart from PSP patients compared to healthy patients. So always these are from volunteers. We have our same methodology actually applies to Huntington's disease as well, which is not quite based on the same protein, but a Huntington's protein, which actually also undergoes the same kind of effect, monomer, oligomer, fibril. And that causes cognitive defects and behavioral changes, motor defects usually. And in this case, we correlated the scores which were derived from this imaging, from this spectroscopic approach, with actual clinical measurements. Currently, clinical measurements are done through questionnaires, behavioral tests, and so on. And we found that actually our method was our laser-based method, which gives results in minutes without any sample preparation, was actually be able to give direct correlation with the severity of the disease. Yeah? And actually, if you see the first point here, which is 0 to 4, which is pre-manifest disease. So actually, it could detect things before the behavioral deficit or behavioral disorders kicked in, before cognitive defects kicked in. Yeah? So with this approach, actually, I've come to the last part of my talk, which will take just a minute. So all of you must know Starship Enterprise and Captain Kirk's crew, right? And one of the very most important crew members was Dr. Leonard McCoy. And he had this tricorder, yeah, which had a small head, and he used to take it around, point it out to different objects, and tell them whether they are dead or alive, alien, human, harmful, dangerous, or not. Yeah? So our vision, honestly, is to develop these kind of devices which can deliver a portable system to the clinic for detection of these misfolded proteins, so that actually we can detect pre-manifest signs of disease right at 20 years before what ROX wants. That's exactly what we want to deliver. And we are somewhere along that road. I mean, early prototypes are being developed in the lab. They look like this, on a breadboard, open. But we want to actually shape them so that they are available for medical researchers like ROX and others to unravel disease mechanisms. We may be able to actually apply them on the interstitial fluid, which ROX is collecting currently or wants to work on. We want to do more clinical feasibility trials of these devices and make sure that these get translated to the clinic within 5 years, and that we have all this translational pipeline all available in Southampton for doing that. And with that, I'll end my talk by giving you a brief summary that actually, coming together, ROX and I were brought together by this Interdisciplinary Dementia and Aging Center, IDEAC, which is an excellent thing for actually making things happen together. And I can tell you from personal experience that interdisciplinary research is one of the most exciting and rewarding areas to work on, because then you can actually see how you will take your fundamental knowledge into the real world and make an impact. Because problems don't see disciplines. They just want a solution. And the solution has to bring people together to actually find the solution. You have to bring people together. So medical researchers and clinicians need novel tools and technologies for understanding disease and finding new treatments, and hopefully we'll be able to contribute a bit to that. But it's best to work with problem owners directly, and patients, of course, to actually develop those tools and technologies. And we are trying to do our best in that area. And I will reiterate once more that only by working together can we actually make a difference. Thank you.
We have come to the end of our time. Many thanks to Sian, Owen, Sumit, Rox, Diana for their contributions here. I hope that some of you have found this thought-provoking, stimulating, even inspiring. Going back to the title, I hope you will reflect that the future of medical research, smart partnerships, is a really good way to progress, and Southampton is doing some really amazing things in this area. Many thanks for taking the time to be with us here. I hope you've had a great time. Now go and enjoy your lunch. Thank you.